All right, hello everyone. Hope you can all hear me. I think we might as well get started. People are still trickling in, but I think it's worth just getting started. So thanks everyone for attending. My name is Ted Natoli. I'm a member of the CMap team at the Broad Institute, and I'll be speaking today on behalf of the team about some updates we've made to our transcriptional resource at the Broad. And of course, if there are questions, please jump in and ask. We'll also have a couple of demonstrations of some of our newer tools throughout the presentation. So just to give an overview, we'll start with some background on CMap and L1000 for people who may not be familiar with our technology and the data that we're generating. We'll talk about the updated links resource that we've generated, which now includes over 3 million transcriptional profiles. We'll talk about some modes of accessing this data, including a migration we've made to Google BigQuery to enable easier access of the data. And we'll have some interactive demonstrations about how we use BigQuery to perform ad hoc analyses, and also a more standard analysis of querying this new data set using tools at clue.io. And as time permits, we can show a couple of vignettes on interesting subsets of this larger data set and talk about some ongoing challenges that we're still working on. So just to give some perspective, the connectivity map at the Broad, we're the Link Center for Transcriptomics. And of course, that's one of multiple centers around the country that are working on building this links resource. And we work very closely, of course, with the DCIC to help share and make available our data. So the general CMap concept is that we're using mRNA signatures of cell state to link genes, drugs, and disease. And so these signatures of cell state are essentially representations of the transcriptional state after something like perturbation or perhaps a disease state. But we use these transcriptional states as sort of fingerprints that represent the underlying biology, and we can use those to find associations between different perturbations or perturbations and disease. And the way we generate our data is by perturbing cultured human cells with various types of perturbations. So these can be small molecules or genetic loss of function reagents like CRISPR or shRNA, or overexpression constructs, or really anything, although those are the main types of perturbations that we use. We perturb the cells, and then we measure the downstream gene expression response. And we catalog those in a large database of gene expression profiles that can then be analyzed in a number of different ways. So to make this resource as useful as possible, we needed to scale up our ability to measure these gene expression profiles. And we imagined that the scale we needed was millions of profiles such that the resource could be as rich and diverse as possible and enable discovery of as many possible associations. And so to achieve this, we used a new assay called L1000, which is similar in spirit to something like a microarray or RNA-seq. But instead of measuring every last gene, we're measuring a reduced representation of the transcriptome, which includes around 1,000 landmark genes chosen to be sentinels for the remainder of the transcriptome. And we find that when measuring these roughly 1,000 genes, we can still capture about 80% of the associations that we would have found had we measured every last gene. Secondly, we use a Luminex-based system to perform these measurements, as opposed to something like RNA-seq or nanostring or microarrays. And so that allows us to achieve an even higher throughput and reduce cost relative to those other technologies. The basic idea is that we treat these cells with perturbations, we collect the cell lysate and capture mRNA, which is then reverse transcribed into cDNA and PCR amplified with a biotinylated primer, which can then be stained with streptavidin stain after it's hybridized to Luminex microspheres, where each of those microspheres is interrogating one of the landmark genes. And so when that bead is read out on the scanner, we get two pieces of information. One is the identity of the bead, which tells us which gene it was measuring. And the second is the fluorescent intensity of that streptavidin stain, which is then proportional to the transcript abundance. And so with this assay, we were able to build a large resource of gene expression profiles. This was made public initially back in 2017, although subsets of it had come out before then. But the data set at that point was around 1.3 million gene expression profiles, comprising around 28,000 perturbations. And this graphic in the center of the page kind of illustrates the breakdown of that data set into two relatively equal sized chunks, one of which we call Touchstone, which is the reference data set, which essentially contains perturbations that we know something about. They're well annotated in some way. So if it's a compound, we know the mechanism or the target, or if it's a genetic reagent, it's targeting the wild type version of the gene, and we know something about the gene, whether it's the pathway or the gene family, so that when external gene sets are compared to our data set, a comparison to something in Touchstone gives you a hypothesis because you're connecting to something that is known. Whereas the other portion of the data set we refer to as Discover, and that's mostly novel unannotated small molecules that come from screening decks or novel chemical synthesis, which is interesting, but not as useful in a lookup sense because nothing is known about those perturbations. And at the time of the release, the data set contained about 80 cell lines. A subset of 9 cancer cell lines were profiled most predominantly and densely, and that comprises the Touchstone portion of the data set. But there were other more ad hoc projects that were done in more niche types of cell models, and that there's sort of a long tail of cell models that have fewer profiles, but in total there's around 80 different cell lines. So in this updated resource now in 2020, there have been additional data generation efforts that have been ongoing for a while, including Links Phase 2, as well as other academic and commercial partnerships that we've engaged in. And so we've been collecting this data, and it's now been organized and is prepared for release currently through a few different mechanisms, which we'll talk about. But notably for this new data set, we've expanded into a few different types of perturbations in cell lines, including CRISPR perturbations, which are a large chunk of the new data set, as well as hematopoietic cell lines and some non-cancer cell lines. So in total, the new data set is over 3 million expression profiles, so it's almost a 3-fold scale up from the release back in 2017. And the plot on the right just gives you a high-level view of one of the metrics that we compute from these signatures. So it's a distribution of signature strength, which is really just the fraction of genes that are significantly up or down regulated in a particular signature. And it's split by the different types of perturbations, from compounds on the top all the way down to CRISPR at the bottom. So this is just a very, very high-level view of the data set, but already you can start to see some trends that particularly maybe CRISPRs are a little bit more subtle in terms of the gene expression changes they induce relative to treatments like compounds, for example. So just a high-level snapshot of one of the metrics, but we can drill in a bit more and discuss specific changes to the data set relative to the 2017 release. So in the new release of data, there's around 82,000 different perturbagens that have been profiled, 33,000 of which are compounds across 657 different mechanisms. And there's about 9,200 genes that we've profiled by at least one genetic perturbation reagent across 240 different cell contexts, 12 of which are primary cell lines, for a total of about 3.18 million profiles, and after collapsing biological replicates, about 1.16 million signatures. And so these plots on the page give you some idea of where increases in the data have been made. So for example, this first plot shows the number of perturbations by type. So we've got about 14,000 additional compounds relative to the release in 2017, and about 11,000 CRISPR reagents, whereas in 2017 there were none. So that's a notable increase there. In terms of cell lines, these are the top 10 cell lines that have, the top 10 cell models that have had the largest increase relative to the 2017 data set. So the biggest increase is in lung cancer. We actually have a lung-specific focused subset of the new release, as well as lymphoma and leukemia, which are part of the hematopoietic subset of our data. There's around 130 or so new mechanisms of action that are now present in this data set relative to the last release. And now we have these 3 different types of genetic perturbations, and there's around 1,200 genes that have been targeted by all 3, and a number more that have been targeted by at least 2. So lots of opportunities, I think, to make comparisons between these different modes of perturbing the same gene. So it's a lot of data, so we needed to think about how to make this data accessible. So for those people who have worked with our data before, you'll be familiar with the GCTX file format, which has been very useful. That's been our workhorse file format, and I think it continues to be very useful for us. But in order to work with a data set of this size, we thought we needed something that's a bit more scalable. And so we've been working on migrating our data to Google BigQuery. So we've got a process in place that will allow us to take our GCTX files along with their associated metadata, load those into BigQuery, and take advantage of things like indexing and a controlled vocabulary, and the ability to slice out arbitrary subsets of our data. And so this data set now sits behind an API and Clue that enables access to the data that way. We also have a Python package called the CMap BigQuery Toolkit, which we'll demonstrate in a few minutes, that enables access to arbitrary subsets of this data. And it has some nice wrapper functions to perform some common operations. So the other sets of tools that we have for accessing this data include web apps on clue.io. There's a data API that I just mentioned, as well as the Python BigQuery Toolkit. The data can also be accessed directly through the BigQuery console. It is a publicly available data set at this point, and we'll have some instructions for how to access that data that we can follow up with after the workshop. And of course, we'll make the data available for downloads from places like the DCIC, our website, clue.io, and Geo. So some example use cases for the data, I think probably the most common one is this question at the top where people ask, what does my gene set connect to? So that's probably the most common use case of CMap data is you have a gene set that you derived somehow, either from doing an experiment or a pathway gene set that is borrowed from an online database or somewhere, and you want to know what perturbations in CMap does it look like. And so that's enabled through our query tool, which I'll show in just a minute. But we've also got a whole bunch of metadata surrounding these experiments. And so common use cases there might be asking what compounds and mechanisms are available in the CMap data set. What are all the compounds that have a certain mechanism? And once you have those answers in hand, you might then want all of the numerical data for that set of compounds across multiple different data levels. Including normalized, differential, and then replicate collapse data, so levels 3, 4, and 5 in our common vernacular. You may also want compounds from a given mechanism and all of their associated data, or maybe slicing the data the other way and looking for the data for across all signatures, but only for a given gene, maybe to identify perturbations that significantly modulate that gene in particular. So all of these use cases are made, I think, more efficient with this migration to Google BigQuery. Before going too much deeper into that, I did want to do a demonstration of our query tool. So this, again, is probably the most common use case of the data. So the question here is, what does some external gene set connect to in CMap? And so the way this works is you would navigate to clue.io/query, and I'll show this in a second. Input a name for your gene set and copy and paste in the genes, and then hit run. And that will take a few minutes to run on the back end, so I won't do that live here, but I'll show some pre-computed results. The query is run against all 1 million signatures. But to make the results a little more manageable to view in a browser, we have a curated subset of about 400,000 that are returned to the UI. But the full results are available for download via the history app in Clue. And so I'll just jump over to that now and show what that looks like. So this is the clue.io query app. This is what the interface looks like. So for example, if we wanted to input a new gene set. We could give it a name, select that we want to run a gene expression query, so comparing against L1000 data. And importantly, we want to select the latest build. So this will enable querying against the entire 1 million signature data set that I just mentioned. Then we would copy and paste in our genes into the up and down box and hit submit. And eventually that result will show up in our history over here. And at that point, we can download the results, or we can view them in a heat map. So I've got a couple of pre-computed results to illustrate over here. So this is what the view looks like after the query has been run. It's essentially a one-column matrix, which is sorted based on strength of association to the input gene set. So in this case, the input gene set was indeed this compound GSK212, also known as trametinib. This is data that we obtained from Geo and did our own differential expression analysis. So this is microarray data. And we're comparing that gene set to all of the signatures in the CMap database. So you can see there's 420,000 rows in this matrix. You can scroll through and identify those perturbations that connect strongly to your gene set. It can also be sorted and filtered or searched in the rows or columns. But I'm just going to highlight the top of the heat map here. These are, the top roughly, let's say 50 connections or so out of 420,000. And you can see there's a very strong enrichment for MEK inhibitors right at the top, and other inhibitors of associated pathway members. So if we didn't know what the mechanism of trametinib was, we could probably guess it's a MEK inhibitor based on the top of this connectivity list. Another example, just to illustrate another strong signal in the data set. This query was the hallmark gene set for NF-kappa B, TNF signaling through NF-kappa B. And we see that at the very top of this list contains a set of interleukins and other cytokine signaling molecules, suggesting that the signal there is pretty strong. And these are some expected associations given what the input gene set was. So these are just a couple of examples that sort of illustrate how these queries would be run. And yeah, as I was saying, this is a curated subset of about 420,000 of the connections. The full list is downloadable via the history app. So that is probably the most common use case for querying our data, but it's sort of a fixed analysis. But I'm going to turn things over in a minute to my colleague Anup, who has worked on developing this BigQuery toolkit in Python, which enables much more flexible access to the underlying data and metadata, and allows you to perform ad hoc analyses thereof. So Anup, I will hand it over to you.
Hey everyone, this is visible?
Yeah, it looks good.
Okay, great. So yep, as Ted was saying, I'm going to be demoing this package called CMap BQ for BigQuery, which is a Python-based package that allows for targeted retrieval of relevant gene expression data from our data set, which we've hosted on BigQuery. So this package is available from pip, so it's very easy to install. It can be installed just by running pip install CMap BigQuery, and we will release this demo notebook eventually with our workshop, so that in the future people can play around and explore with these tools and see how these analyses work. So just running through this quickly, we have some standard imports for pandas and NumPy and some visualization packages. And there's a little bit of credentials set up. So we have a little bit, it requires a credentials file from Google BigQuery. So we will also release some information about how to set up the tool. Essentially, we need to get a credentials JSON file, and it's as simple as running this setup credentials function one time, which will essentially install all the, will point the toolkit towards your credentials file. And then from here, we import our package here. This is the standard way that I've used it. And then we instantiate a BigQuery client. So this BigQuery client is important because it's sort of pre-authenticated and will be passed into a lot of the applications that allow us to query our tables in the future. So right here, this is just an overview of how our data is organized in BigQuery. I won't go through all of it, but quickly, the level 3, 4, and 5, those are all numerical data, so that includes gene expression values. And then instinfo and siginfo are metadata that correspond to each of these. So instinfo corresponds to the profiles, which are level 3 and 4, which is usually like individual wells. And then siginfo maps to signatures, which are replicate collapsed mod z-score values. And then some of the other tables include information about compounds and how they map to various MOAs and targets, and cell information such as cell names and cell lineages. So what we wanted to do with this demo is essentially show a quick example workflow that might be used to do a quick analysis of an MOA of interest. So quickly, we created a few utility functions such as list CMap MOAs or list CMap targets. These allow us to just quickly see what MOAs are available. This will list all the MOAs available in the resource, similarly with list CMap targets with targets. And this is useful to ensure that your MOA of interest is present in the data set and the resource before running further queries. So given that now that we know that we have one MOA of interest available, we've listed this glucocorticoid receptor agonist as our example MOA. So one thing we may want to do is find out, get some information about what compounds may relate to that MOA. So we do that using the CMap compounds function here. That queries the compound information table. So the way this is run is we pass it that BigQuery client object, and then here we can filter by MOA. So this is an MOA keyword argument. And we are passing it that MOA glucocorticoid receptor agonist. So this is a sample of what's returned, just the top 10 rows. And here you can see all of the outputs are glucocorticoid receptor agonists. So there's one row entry per CMap name, target, and MOA combination. So there are times you may get multiple of one CMap name, but multiple targets. So there's one per combination. So another thing we may look for is cell line information. So I've listed here the 9 core cell lines that Ted briefly mentioned before. These are just perhaps the most abundantly available for signatures. So we chose these, and here we're querying the cell info table. So this is queryable via the CMap cell method. And again, we're passing the client, and then we're filtering on cell iname, and then passing a list of cell names, right? And here I've also included this verbose true flag. This is more for like more power users, or if you want to use the console in the future, this will show the actual query that's being run behind the scenes. So this is the output of that. This is the cell info table where we have various columns related to the name. Things like growth rate are available. Cell lineage is also something that is available and quite useful. So now that we have some MOA compounds related to the MOA, and we know a few cell lines that are related to that or that we're interested in, we now want to find some signatures that match these filters. So the SIGINFO file provides information on signatures such as like time point, dose, compound name, cell line, many experimental parameters, but it also includes some signature quality metrics such as signal strength, the number of genes modulated, replicate correlation, and our transcriptional activity score, which is an aggregate measure of both the strength and the reproducibility of a given signature. So to use the tool, in order to get that, to query the SIGINFO table, here we use CMap query.cmapsig, which queries that table, again, passing the client. And then we're passing sample compounds, which is a list of the compounds related to this MOA. Sample cell lines, which are the cell lines mentioned previously. And then that returns a sort of full list of every entry that has a compound within this list of compounds and a cell line within this list of sample cell lines. And then from there, it's pretty useful to filter it a little bit further. So here we filter for signatures that have 3 or more samples. So this is replicate collapse signatures. So this is, we're filtering for signatures that had 3 or more replicates going into it. We checked that dose was greater than or equal to 10 micromolar, so a higher dose, and we selected one time point for this example. So here's again a sample of how the SIGINFO outputs a multitude of different metadata information. And before we go straight to the numerical data, it is important to point out that this SIGINFO table is quite useful in and of itself. Our transcriptional activity score is something that we use pretty frequently to screen for bioactivity, often for example, collaborators, or when preparing as like a pilot screen for larger screens, we tend to use transcriptional activity score to get a sense of what types of experimental parameters are ideal, or what sort of cell lines may be interesting. So this code here is essentially just plotting. So we've plotted this box plot of the transcriptional activity score based for each of these cell lines. And it's been colored by the cell lineage. So here we can look for trends. In this case, we see that lung cell lineages seem to have the highest transcriptional activity score, which is expected for a glucocorticoid MOA, as lung cells are where that receptor is most highly expressed. Right? So from here, now that we have these these signatures that we want to extract, we can query the numerical data to get the raw gene mod Z scores for each gene for these signatures. So the signature ID or SIG IDs are sort of the mapping between the SIGINFO file and the numerical data. Each SIG ID is unique for each row in that SIGINFO table and maps to the numerical data. So we want to collect all the signature IDs, and then we pass it to the CMap matrix function, and we'll pass it as CIDs, so column IDs. So here in this data set, columns are signatures and rows are various features, which in this case are genes. So after we run this query, it'll run the query. It'll take varying amounts of time based on how many signatures you're interested in. And I've printed out the shape of the resulting data set. So this is 978 genes, which is our L1000 space. times the 120 signatures that passed our filters. So this returns a GC2 object, if you're wondering why it's this .data.df attribute. That's part of our CMap Pi resource and is related to the GCTX file format. And there's documentation on how that's structured if you're interested in that. So from there, we can, of course, write that file as a GCT or GCTX file, which we can view in some other tools such as Morpheus on Clue. But for this demonstration, we wanted to show what we can do with something like pairwise correlations. So we took this data set and just created a correlation matrix between all of the signatures. And then we subsetted that correlation matrix by cell line and then plotted, firstly, a histogram of all those correlations. So this is just an example for how we may look at what cell lines provide the most concordance between within a certain MOA, right? So here it's sort of, it's ordered by median correlations, and we can see that on the bottom right are actually two lung cell lines, which show the highest pairwise correlation or highest median pairwise correlation between all signatures in that cell line. This essentially can be condensed into a box plot as well. So this is the box plot that's showing very similar data to the histograms. And we can see again, these two lung cell lines are available, which is good that L1000 can show this, that behavior, that expected behavior. And then finally, we like to view these as heat maps. So splitting this, these correlation matrix by heat maps, we're able to generate these views, which are nice visual representations of what compounds actually like tightly cluster together and are highly correlated. So because this is all of one MOA, ideally you would like to see a strong block if it is if that MOA is well characterized or well represented in this cell line. So again, we see that in the two lung cell lines, and maybe see it less prevalently in some of the non-lung related cell lines or other cell lines available, right? So this was, this generally was just a view of how you can pull specific data related to one MOA, and then do a few different types of analyses and maybe generate some hypotheses from just from the raw gene modulation data available on BigQuery. So now I'll hand it back over to Ted, and he'll go through some interesting characterizations of the data.
Okay, thanks, Anup. Moment. All right. So we just went through a couple of common data access modes. And there were some questions in the chat about when these tools would be available. So just to clarify, we'll be following up after the workshop with links to that notebook that Anup just illustrated, along with some instructions for how to set up your Google account so that you can access our data set. This query app will be moved over to the production clue.io website very shortly after the workshop as well. So all of these things will be available very shortly following this workshop. Right. So now that we've talked a bit about how to access the data, I thought it might be useful to highlight some potentially interesting subsets of the data that could prove useful for analysis. So these subsets include compounds that we've profiled at dose, as well as CRISPR signatures, a subset of hematopoietic cell lines, and ligands that we showed in the query example. So the main, I think, the overarching idea here is what questions can we ask of this data? So we'll show a couple of example questions that we can ask for each of these data subsets, but I think it's worth just thinking about what other things might be able to do with these data sets. So the first of these data sets is a subset of drugs that were profiled at dose. So for people who are familiar with the previous release of the data, they'll notice it was mostly done at a single dose, typically 10 micromolar, and there were very few compounds that were profiled systematically at dose. So there are subsets of the new release where we have done that dose profiling. So one example is about 1,500 compounds that have been profiled at a 6-point dose series in 3-fold dilutions down from 10 micromolar. And that have been done in 10 cell lines. So it's a pretty dense subset of the data set where this data has been made systematically. So some questions we might ask of this are how might we identify a dose response if there is one? And that can be done using transcriptional activity. And how often do we observe such types of dose response, and what types of responses exist? So we've taken a first pass at trying to answer some of these, but it's useful for others to think about how they might do that as well. One useful metric for analyzing this data is the Transcriptional Activity Score, or TAS as we call it. So this is one number that tries to give us a sense for whether the treatment did anything in the cell lines. So TAS is a metric that's comprised of two things. One is the strength of signature, so that's the number of genes that were significantly changed, and then the replicate correlation between the biological replicates that comprise the signature. And so we take a geometric mean of those two numbers and scale it so that it ranges between 0 and 1. And this plot on the left shows those strength versus correlation metrics plotted for all of the compounds, a large chunk of the compounds in our data set, and then the DMSO vehicle controls. And we see that the compounds, of course, are much stronger, and that's as we would expect. And so we can sort of calibrate a TAS threshold based on the negative controls. And we find that rarely, about 5% of the time, do negative controls exceed a TAS value of 0.2. And so that's a sort of a heuristic threshold that we use to identify active signatures. If they exceed that value, then we think they're significantly more active than the majority of the DMSO population historically. And this plot on the right with the isobars indicates where what strength and the replicate correlation values would correspond to a particular TAS value. So one way that we can characterize dose response is using this TAS and just simply plotting the TAS value, which is shown on the y-axis here, versus the log of the dose on the x-axis. So this is a particular compound, which is a CDK inhibitor, AZD5438. And we can see that as the dose goes up, the strength of the signature, the activity of the signature also goes up, and that happens universally across all the 10 cell lines. We can quantify the degree to which there is a dose response by doing something simple, computing a linear model and fitting that to this curve, and then using the slope of that model to as a proxy for the degree of dose response, and then the median TAS value overall as a proxy for how strong that signature is in general. And so if we do that across all these compounds that we've profiled at dose, we can then compare that median TAS value on the y-axis versus the slope of that linear model on the x, and sort of coarsely bin the compounds into these various different categories. So in the upper right, this is a compound where there was a high slope and a high strength overall. So this would be a strong compound that was dose responsive. There can be other compounds that are dose responsive where the slope was, there was a good linear model fit with a positive slope, but the overall strength was low. So there's some dose response happening, but it's a subtle signature. Other cases, and this is where most of the compounds fall, in the inert category, where there's pretty low TAS and not much of a dose response. Another pattern we see sometimes is a dose-saturated response, where there's basically the slope is relatively flat, but all the points are high. So even at the lowest dose, we're eliciting the strongest response for that compound. And this plot at the right shows the distribution of those dose-response categories across these 1,500 compounds. These are some examples of dose response and compounds that have different patterns of response. So here is the CDK inhibitor that we showed earlier, which is dose responsive across all cell lines. This compound, norethisterone, is selectively dose responsive only in HeLa cell lines, which I think sort of makes sense based on the mechanism. Bortezomib, a proteasome inhibitor, is broadly dose saturated, so it's having a very strong response across all cell lines, and that doesn't really change with dose. And then fulvestrant is another compound that is an estrogen receptor antagonist, and that has a dose saturated response, but only in MCF7, as we might expect. So you can kind of use these views to identify different interesting patterns with these compounds that have been profiled at dose. A second interesting subset of the data is the CRISPR data set. So this is, again, 5,000 genes that have been profiled across 10 cell lines pretty uniformly. And we've done these at mostly 2 guides per gene, although some have a few more, but the majority of them are done with 2 guides per gene. So one question we can ask of this data is whether CRISPR results in reduced expression of the targeted gene. And how do CRISPR signatures correspond with shRNA and compounds? And this plot on the right just kind of gives a sense for the diversity of genes represented in this CRISPR data set by mapping them onto Panther pathways. So you can see there's a large number of different pathways that are represented to some extent. So it's a fairly diverse representation of genes. So to answer that first question, we can look at the CRISPR signatures that we've generated where we've targeted a landmark gene. So this is a gene this is one of the 1,000 genes the assay is directly measuring. And we can ask, when we target that gene, does its expression level go down? And we can assess that by looking at the Z-score for that particular gene in the signature. And so we've set up some heuristic thresholds to assess whether a gene was significantly downregulated. And so our criteria for that is that the Z-score is less than -2, and the rank of that gene across all samples on the same plate is within the top 10 in the sample where it was being targeted. And so these are distributions, cumulative distributions for these Z-scores and ranks in the 10 different cell lines, stratified by whether the particular guide we're using is a test guide in blue or a reference guide, which is a known high quality, highly effective guide in red. And so overall, we see that around 60% of the genes that we targeted were knocked out in any given cell line. So that's certainly a non-trivial fraction, and I don't know that it's necessarily a given that a gene that is targeted for knockout would necessarily have reduced expression, but we're seeing that more often than not, that is the case based on our data. Another view of this data is to look at the transcriptional activity score for all of these genes in all of the 10 cell lines. And so that's what this heat map on the top represents, where the intensity of the cell in the heat map is proportional to the TAS of the particular gene. One thing you can notice is that it's fairly sparse. Most of these knockouts don't induce very strong signatures, but there are some subsets that do, and it winds up in many cases being very cell line dependent. So there are some examples here below where estrogen receptor knockout is really only giving an active signature in MCF7, and BRAF knockout is only giving a strong signature in BRAF mutant cell lines, which I think makes sense. There are other genes which are more context agnostic, such as MYC and this proteasome subunit, where in almost all of the cell lines, you get a strong signature by knocking out that gene. So we do see some differences in those patterns, but overall, I think it's worth noting for people who plan on working with this data, the signal is fairly sparse, and it can be quite dependent on the particular cell line. And one thing we've noticed is that the baseline expression of the gene is very important in determining whether you get an active signature in the particular cell line. So that's something that's worth keeping in mind for folks who plan on working with this data. One other question we can ask is comparing signatures of CRISPR knockout and shRNA loss of function to compound signatures that are directed at a particular gene. So here the question is, how often do we see an association between a compound signature that targets a gene and then a genetic loss of function of that targeted gene? So the plot on the left is the distribution of the connectivity scores, or tau values, between compound gene signature pairs, where the data have been stratified based on whether the genetic loss of function signature was derived by shRNA knockdown in blue or CRISPR knockout in red. And so one thing that we can see in both distributions is there's a nice right shift to that distribution, and there's a spike at very high tau values, which is good. So this suggests that in general there's more similarity than not between signatures of compounds that target a gene and genetic loss of function signatures of that gene. So that's good. The plot in the center shows a comparison of the aggregated connectivity for a compound gene connection between those connections made using CRISPR and those made using shRNA. And so you can see, there is an enrichment of plots in this upper quadrant, which sort of makes sense based on the plot at left. So generally, these associations tend to be positive, but there is quite amount of spread here. So it's not always the case that the genetic loss of function signature from CRISPR and shRNA track in the same to the same degree when compared to a compound signature. But if you apply some thresholds to these data, where we're asking for the aggregated connectivity between a compound signature and the genetic loss of function signature, if that exceeds a tau value of 95, meaning that the targeted gene signature is in the top 5% of all connections to the compound, we'll consider that a successful recovery of the targeted gene signature. And so of this data set, where we've got 358 genes that are targets of compounds, and we've profiled them by both shRNA and CRISPR in the same cell line, of those 358, 87 connect successfully by shRNA and 62 by CRISPR, so a slight difference there. 41 by both, so there is some enrichment, I think, for commonality in this very upper right portion of the plot. But there's 108 that connect by either. So I think that that's good. That gives us 108 signatures of these genetic loss of function signatures that we believe are good, because we're able to match them to a perturbational signature of a compound targeting that particular gene. And it's not always the case that CRISPR and shRNA give you the same answer, but it's nice that using both of those methodologies, we can then increase the coverage of genes for which we believe we have a good loss of function signature. So another subset of interest that I'll touch on is the hematopoietic subset of the data set. And so this is data that we made in conjunction with the biotech company H3 Biomedicine up in Boston. And this is 140 drugs that were tested in 11 hematopoietic cell lines, a large subset of which were DLBCL cell lines. And these were profiled to identify the differences in response we might see between the different subsets of hematopoietic cell lines, particularly ABC and GBC subtypes of DLBCL. And so some questions we can ask of this data are which mechanisms are active in these heme cell lines, but not in the solid tumor cell lines in our previous reference data set, and whether L1000 can distinguish between these DLBCL subtypes. So we focused primarily on the BTK inhibitors for these examples because they do give quite a stark difference between hematopoietic and solid tumor cell lines, as one might expect, based on where these genes are expressed. So BTK itself is largely only expressed in hematopoietic and lymphoid tissues based on looking at data from CCLE. This is across around 1,000 cancer cell lines, and it really is only expressed in this one lineage. And so then if we look at the connectivity of BTK inhibitor compounds to each other, so we're asking as a class, how well do these BTK inhibitors recapitulate each other's signatures? How tight of a cluster do they give in each one of these cell lines? So this is a radar plot where each spoke is a cell line, and the distance from the origin is proportional to the strength and coherence of that BTK inhibitor class in that particular cell line. And so we can see there's a stark difference between the hematopoietic cell lines and how strong the signature is and how coherent that class is in those cell lines compared to the solid tumor cell lines from our reference data set, where in most of them, the class does not give a coherent signature. So notably different, which is good, and this sort of tracks with the corticosteroid example that Anup showed in his demonstration earlier. We can drill into this a little bit further and ask, is there not just a difference between solid tumor and hematopoietic lines, but within the hematopoietic lines, can we distinguish the subtypes of DLBCL? So this plot at the left is a PCA projection of all of the DMSO samples from this 11 cell line data set, colored by the DLBCL subtype. And so we can see that the activated B cell ABC subtype in red and germinal center B cell GBC subtype in green, they do separate along these two principal components, which is nice to see. So there is some distinction just based on the 1,000 landmark genes between these subtypes of DLBCL. And if we go back to looking at the BTK inhibitor class again, and now we're just restricting our view to the hematopoietic cell lines, but recoloring the points based on the DLBCL subtype, you can see that the BTK inhibitor class is notably more coherent in the ABC subtype of DLBCL than the GBC. And this does track with the clinical observation that ABC patients tend to be more responsive to BTK inhibition than the GBC patients. So it's nice that L1000 is able to recapitulate this clinical observation in our data. So I want to give some time for questions at the end, but just looking forward, there are some things that are still open challenges and things that we're thinking about and would love input from the community on. And perhaps, you know, the biggest of which is how do we distill information from this long list of query results? So now, you know, we've expanded the data set about threefold. So this problem has only gotten worse, I think. But, you know, when you run a query, you get a long list of results. And we showed some examples today where the answer is very clear. There's sort of a very strong mechanism right at the top. But in many cases, that doesn't always play out so cleanly. So I think it will be important for us in the community to think about how best to, you know, to extract some information from that long list. So things we've thought about and things we've tried doing in the past are summarizing across cell lines. But in some cases, those cell lines don't overlap. For example, the CRISPR data and the compound data that we're releasing only contain a small number of cell lines in which we have data for both of those perturbogen types. So being able to kind of summarize in a uniform way across cell lines is not necessarily possible. So we'll need to sort of think about how best to do that. Other things we've thought about are integrating data from different perturbogen types. So we showed comparisons between shRNA and CRISPR. But I think it's worth considering whether there's a way that can be formally leveraged to help glean some more insights from these long lists of query results, as well as leveraging some redundancy in the data. You know, for example, we have many compounds that have been profiled more than once, maybe at different doses or at the same dose, just on multiple different experiments. So how do we use that multiplicity of signatures to help increase confidence in a connection? So these are all things that are worth thinking about, and it'd be great to have input from the community as well. So I just want to conclude by thanking everyone, especially on our team, Arvind and Todd, who provide our leadership, Rajiv, Jacob, and Anup, who've helped with analysis, John and Sarah and Veed, and previous members of the group, Dave, Nick, Andrew, and Juana, who've helped with data generation or analysis for this work. And I also wanted to thank folks at the DCIC, especially Dusika, who's been very helpful in getting all of our data wrangled and imported into their system so that it can be shared with the broader community. And before ending, I just also wanted to mention that there is going to be another upcoming workshop focused on CMap links in L1000. This will be December 17th and 18th. I think there should be invites that have gone out or will be going out shortly. But this will be more interactive. I think we'll have several more sessions that will focus on tools for data access and analysis, and hopefully can build on some of the points we've touched on today. So it'd be great if folks could sort of start thinking about that and thinking about questions to ask, and yeah, we look forward to seeing you all again at that point. But thank you, everyone. Appreciate everyone attending, and I'm happy to take any questions. We have some questions coming in through the chat, so I can just try to verbally answer those. Yes, we will definitely post the slides. Those can be shared, absolutely. And there's a question about, are there any future plans on generating data across organoid-based screening systems, 2D versus different microenvironments, preclinical systems? So I think we've done some very early exploratory work on that. I don't know exactly what the status there is. So I don't think there's immediate plans for that. It's very exploratory at this point. So I can't say definitively, but it is something that we've thought about. So it may be in the works in the future, but I don't have anything specific I can say beyond that. There's a question about when will the hematopoietic data set be available on Clue. So very shortly. So after this workshop, probably in the next day or two, we should be posting this entire data set, including the hematopoietic subset on Clue. So the whole thing should be available there very shortly. And that same goes for the CRISPR data set as well. So this whole data set, CRISPR, compounds at dose, heme, all of those things, that will all be available on Clue within the next couple of days. Question about the registration link for the December workshop. Yes, we can send that out. I don't have it handy with me at the moment, but we should be able to, yeah, we should be able to, through links, disseminate that to everybody who attended this workshop. So we can make sure everyone gets that link. Oh, I take it back. Well, I guess we do have it. So if you go to, let me just post it in here. I'll just Slack this to everyone. So if you go to clue.io/workshop3, I guess that is the link to register. There's a question about office hours. Yeah, we still do host office hours on Tuesdays. So for folks who have questions, we have a Zoom office hours every Tuesday at 1 p.m. If you visit clue.io/office hours, there's the instructions for how to log in there. But we do host that every week. Someone's asking whether it's possible to download the shRNA data now. Yes, it is. So that is available at clue.io. If you go to the clue.io/data, the data library app, there's an entry for Touchstone there, and that contains all of this, all of the data from the 2017 release. It's also available in Geo, but if you just go to clue.io/data, it should be very prominent there, so you should be able to find it. There's a question about whether we have multiple myeloma cell lines. I believe So in that hematopoietic cell line data set, there were, I think, 4 myeloid cell lines. I don't know if they are multiple myeloma explicitly. I could check on that, but we have all the, we can share the, all the annotations for this data will be available too. So the cell line names will be accessible. So for folks who are more familiar with the heme field, they may recognize those cell lines, but that all should be available.
Ted?
Yes.
Hi, can I ask a question?
Sure.
Can you share your thoughts about analyzing upregulated and downregulated genes separately as opposed to all together as a signature? Would it lead to different results? I'm dealing with this right now, so I just want to maybe your opinion.
Oh, sure. Yeah, I mean, I think it can sort of depend on the mechanism. We by default tend to use both sides of the signature, but there may be cases where some perturbation or some transcriptional state, really only one of the 2 directions matters. So I think it kind of depends. Are you using CLUE to do these analyses?
Yeah.
Okay, and are you getting very different results when you run a one-sided query versus the 2-sided?
Well, in general, I get more fuzzy data if I use everything all together, and many more hits using one side of the of the scale. But then it's hard to prioritize, I guess, in a way. So I'm just thinking, yeah.
Well, you may look at your how you're generating these gene sets could be influential as well. So for example, if you're doing some kind of differential expression analysis and you see that the fold changes for upregulated genes are notably larger than those for downregulated genes, maybe that tells you that the upregulation is a more prominent effect in whatever it is that you're studying. So you could use that to help kind of guide your analysis, but I don't know if that's something you're currently facing or where you're getting these gene sets from. But you might look at the input data before you actually run the query to kind of help guide you in that regard. Okay,
yeah, that makes sense. I try in doing, because I have it's slightly over 150 genes, so I'm ranking them by fold change.
Okay.
And yeah, but I was just wondering about the up and down thing. But
yeah, I think if you're running a one-sided query and you see that the results are much more stark in one direction versus using both, that might make sense to go with that one direction. I coupled that with looking at the input data, if there's any clue there as to which direction is more important. That might be useful. You could also, if you have replicates in your data, you could shuffle the replicates or permute the labels to see whether there is any significant effect in which genes wind up being differentially expressed, and if those changes are more prominent in one direction versus the other, that might also help inform which direction is the more important one.
Okay, yeah, that's some interesting thoughts. I'll take that in consideration. Okay. Thanks, man.
All right, thanks very much.
Just a quick question, please.
Sure.
Yeah, that was a wonderful presentation. But I just want to say that for the clear, if you want to impute the data, because I got confused at another point, if you impute the upregulated gene in the upregulated colon and the downregulated gene in the downregulated colon. So how do you look at the heat map? Do you look at it from the top or do you look at it from the bottom? Because I know there are different rule of thumb. If you interchange it when you are uploading it, there's a way you look at the result. And if you just upload it directly, like put in the upregulated in the upregulated colon and the downregulated genes in the downregulated colon. Do you look at it from the top or you look at it from the bottom?
Yeah, you would look at it from the top. So the convention is that a positive connectivity score means that the reference signature is mimicking the pattern of gene expression that your query reflects. So that means that it is upregulating the genes you put in the up box, and it's downregulating the genes you put in the down box. So you want to look at it from positive to negative.
Okay, then that means you look at maybe probably the first few genes, looking at it from the top, right?
Yeah, I think you'd want to find positive associations.
Yeah.
Right.
Okay. Then about the office hours on Tuesdays, they're requesting for this passcode that wasn't given. Because I've had a couple of weeks that I've tried to join the office hours, and it was requesting, asking me to put in a passcode for the Zoom, but there's no passcode anywhere on the website.
Oh, I'm sorry about that. That should have been fixed.
Oh, okay. Okay.
Yeah, let me double check that. If anyone else, yeah, it looks like a few other people have mentioned that in the chat. So sorry about that. I will double check.
No.
It looks like it might be an issue with the link on our office hours page. We just need to fix that.
Okay, thank you.
All right, I think we're probably out of time. Thank everyone very much for joining and for the participation in the questions. If there's any questions that were not answered, I believe our contact information should be shared in the agenda. So, I'm happy to follow up with people separately.
Just a naive question. Right, Ted? Hello?
Yeah, is there one more question?
Yes, just a naive question. So I was wondering whether tomorrow's, because in the hands-on session tomorrow, there is also another transcriptomics session. So I was wondering whether it will be the same or different material and different perspective on the links.
Yeah, yeah, I think that will be, there'll be some of the same material because it's, yeah, it's sort of based on the same principles that were discussed today, but there are lots of different perspectives and different ways that we've been using our data, integrating it with other types of high throughput data sets. So the presentation will not be identical to this. It will be different material. Some of it will overlap, but it'll be largely different.
Okay, thank you so much.
All right. Sure. Okay, it looks like we're getting bumped out. So thanks everyone again for attending. And yeah, I'm happy to follow up with people separately if there are still questions. So thanks very much.